Hi folks, welcome back to AP Biofun with Dr. D. This is the second part of our video tutorial on proteins, how structure determines function. And I would like to remind you that last class, in the last video, we talked about secondary structure and the two main forms of secondary structure, an alpha helix and a beta plated sheet. You can see it in this slide. To the right is a helix, and then to the left is a beta plated sheet. Now the alpha helix and a beta plated sheet are secondary structures that form because of hydrogen bonding between the backbone of the polypeptide. Remember that, hydrogen bonding between the backbone. R groups are not involved here. And some amino acids are more likely to form an alpha helix, while others are more likely to form a beta plated sheet. And we discussed it last class why that is. That is in part because of the size of the side chains. And not all side chains can fit everywhere. But it's also important to remember that you can have a large polypeptide that has many different regions of folding. So secondary structure is local. Only a small part of the polypeptide will form a beta plated sheet. So if you see here, this is a whole polypeptide and one part is a beta plated sheet, another one is an alpha helix, and so on. So this is local folding. Now, when we talk about the whole protein, we're talking about the 3D structure of a protein. We're talking now about tertiary structure. And tertiary structure is the whole molecule folding, and now it involves the side chains, the R groups. We are no longer talking about interactions between the backbone. Now we're talking about interactions between the R groups or the side chains of the amino acids. So in addition to that, it is important to remember that these interactions can actually involve amino acids that are quite far apart in the polypeptide chain because of that local folding that we talked about in the last class. And the interactions between the side chain could be hydrophobic, as shown here in that pink square, right? So the cytoplasm is water-based, we know that. So hydrophobic, so non-polar amino acids that have non-polar hydrophobic side chains tend to cluster together and away from water. They tend to bury themselves inside the protein and away from the water-based cytoplasm. We also have hydrogen bonds and ionic interactions, and a hydrogen bond is shown here in blue between two polar side chains, and then there's an ionic bond or ionic interaction between two charged R groups right here at the bottom. And finally, we have disulfide bridges. Disulfide bridges are the only covalent bonds that are involved in protein structure, in higher order protein structure. They form when two sulfhydryl groups of an amino acid called cysteine are oxidized and form a covalent bond between the two sulfurs. All right, here is a model of a 3D structure. And in yellow, you will see beta sheets. In magenta, you will see some alpha helices. And what you can see, so this is a whole protein. It's a real protein and it's 3D structure. You can see that a 3D structure, the final native structure of a protein, involves interactions between the secondary structures. The alpha helices, the beta plated sheets, and a protein has a varied number of beta plated sheets and alpha helices, and they also may be positioned in different portions of the protein. And when they come together and fold, you have your whole 3D shape. Okay, so let's examine the R groups, the side chains of the amino acids, because they're the ones that are responsible for these interactions. And here, what you will see is you will see the side chains of several amino acids marked in red. So again, I want to point out that you will see that what is not in red really is identical for every amino acid. So just look at it and compare them. All of these amino acids have an identical kind of a backbone, and what's different is only the part that is in red, which is the side chain. And what you will notice here on these side chains is what? They're full of hydrocarbons, CH3, CH, CH2, CH2, CH, CH3. They're just carbon and hydrogen. And remember, carbon and hydrogen form nonpolar covalent bonds. For that reason, these side chains are nonpolar. Because they're nonpolar, they're hydrophobic, okay? So if they're nonpolar, then they're hydrophobic. And if they're hydrophobic, what kind of interactions can these amino acids participate in? They can participate in hydrophobic interactions. These type of amino acids are polar, but they're not charged. Now, why are they polar? Let's examine the red again. So in red are the R groups, the side chains. This one has a hydroxyl group. This one also has a hydroxyl group. This one has an amino group and an aldehyde group, or a keto. I don't know, what are we gonna call this? Both of these are polar. So the carbon forms a polar covalent bond with the oxygen, and the carbon and the NH2 group, we know it's a polar amino group. So an amino group and an aldehyde group, both of them are polar. So the whole side chain is actually polar, even though it's not charged. This one also has the same type of structure, an amino group and an aldehyde group. So both of these are also polar. These two are polar because of the hydroxyl groups. These two are polar because of the aldehyde and amino groups. How about this one? It's a ring structure, which is only C and H. So this is highly non-polar or hydrophobic, but then we're just stuck to it, a hydroxyl group, and hydroxyl groups are polar. Therefore, the whole R group now becomes polar. Polar R groups can form hydrogen bonds. Remember, polar groups can form hydrogen bonds. Now let's look at the charged R groups. There's two types of charged R groups, acidic and basic. Acidic groups are ones that increase hydrogen ion concentration. They increase hydrogen ion concentration, if you remember from the functional group lecture, because they lose a hydrogen ion and therefore end up being negatively charged. So the carboxyl group here in these two amino acids loses its hydrogen and becomes negatively charged. These three amino acids are considered basic because they have an extra hydrogen here, here, and here. And now they're positively charged. So they have picked up a hydrogen ion from the solution, which means they're reducing hydrogen ion concentration, which means they're basic. So what kind of interactions can these groups participate in, right? What kind? These two are negatively charged, these two are positive, these three are positively charged. So a negatively charged amino acid can interact with a positively charged amino acid, forming ionic interactions or ionic bonds. And I would be remiss if I don't mention these three weirdos. Now, these are amino acids with somewhat of unique properties. The first one is called glycine, and it is hydrophobic. Its side chain is a hydrogen. There's nothing polar about it. It's a single atom. So yes, it is hydrophobic, but it's also the smallest amino acid. Why is it the smallest? Because it has the smallest side chain. Its side chain is a single hydrogen atom. So because of its size, glycine often resides at sites where two polypeptides have to come in close contact with each other. So basically the size of its side chain is not in the way. Cysteine contains a sulfhydryl group. So sulfhydryl groups are polar, but usually cysteines within a protein participate in disulfide bridges. So this cysteine is going to form a covalent bond with another cysteine, which might be very far away. The two H's are going to be removed, and you're going to form a covalent bond between the two sulfurs. So it's going to be a disulfide bridge, the only covalent bond which holds together tertiary structure of proteins. And proline is this weird little amino acid, which is hydrophobic because its side chain is simply CH2, 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 and kind of a ring structure. But because of that weird ring structure, proline it's really weird. It induces bends and kinks in the polypeptide chain. So if you have proline somewhere, chances are you're not going to have a very highly ordered secondary structure. So what is quaternary structure? Well, quaternary structure is when the functional protein really is made up of more than one polypeptide chain. So you can have a functional protein that is made up of a single polypeptide chain. So not all proteins have quaternary structure. Or you can have proteins that, in order for the protein to be functional, you have at least 2 polypeptide chains coming together. Sometimes you can have 3, you can have 4, sometimes more. So how are these 2 polypeptide chains held together? They're held together the same way that tertiary structure is held together. It involves the R groups and it involves the same type of interactions: hydrophobic interactions, hydrogen bonds and ionic bonds, and disulfide bridges. So tertiary and quaternary structure are made possible by the side chains, the R groups. So the R groups interact and the type of interactions are identical. What's different about quaternary structure is that the interactions are between different polypeptide chains, whereas tertiary structure, the interactions are within the same polypeptide chain. Okay, here are some examples of quaternary structure. Collagen is a protein that is a major component of our skin and also tendons. And it is made up of 3 different polypeptide chains. So that's quaternary structure. Hemoglobin is the most famous protein that has quaternary structure. It is made up of 4 polypeptide chains, 2 alpha and 2 beta. So 2 different, but in total 4 polypeptide chains make up one functional protein that carries oxygens through our body. So in addition to, so what determines protein structure? How do proteins fold really? And what determines what the protein is going to look like in 3D? Well, it is the primary structure. So how the protein is going to fold is really written in the sequence of amino acids, or the primary structure. And the sequence of the amino acids is written in our genes. However, proteins may fold correctly, they may fold incorrectly, they may partially unfold, or they may completely unfold, depending on the environment they're placed in. So physical and chemical conditions can affect the structure of a protein. So changes in pH, changes in salt concentration, changes in temperature, or other environmental factors can cause a protein to partially unfold, partially unravel, or completely. It could be partial, it could be complete. It depends on how harsh the environment is. And you know what that is called. You've learned this in bio, it's called denaturation. Denaturing a protein means that this protein is no longer biologically active. Okay, so let's explore briefly, how would temperature disrupt the shape of a protein? You know that if you boil a protein, the protein is going to unravel. If you boil an enzyme, the enzyme's not going to work. If you put an enzyme at pH which is not normal for that enzyme is going to denature and not going to work. So how is that achieved? Well, let's remember that 3D structure of proteins, whether it's tertiary or quaternary, are held together by these type of interactions: hydrophobic interactions, ionic bonds, disulfide bridges, and hydrogen bonds. Temperature, high temperature, is going to disrupt hydrogen bonds. Hydrogen bonds, remember, are weak bonds, so they will be disrupted if you have higher kinetic energy. Some hydrophobic interactions will be disrupted as well at high temperatures. Ionic bonds will be disrupted if we disrupt the balance between hydrogen ions and hydroxyl ions, if we change the pH. If you have too many hydrogen ions, they're going to interfere with these interactions. So, for instance, this carboxyl group may preferentially be attracted to a hydrogen ion rather than this NH3 group. So this interaction is going to be disrupted if you have too much hydrogen ions or too little hydrogen ions as well. If you dissolve salts, remember salts are ionic compounds, so you're increasing the concentration of certain ions in the cell. They also will disrupt those ionic bonds in those interactions. They can also disrupt hydrogen bonds, and the pH, changes in pH can also disrupt hydrogen bonds. Now, how do we disrupt disulfide bridges? Thus, disulfide bridges can only be disrupted by certain chemicals. So chemicals that are able to reduce this, reduce means adding hydrogens to it. Oxidize is the opposite, removing hydrogen. So remember, this is a sulfhydryl group. So originally it was SH. When it was reduced, it formed a when it was oxidized, it formed a disulfide bridge. When you reduce it, you will disrupt this disulfide bridge. So chemicals means chemical reaction. A chemical reaction needs to happen to disrupt this covalent bonds. Well, guess what? Those chemicals are used together with heat, right? Because remember, temperature can disrupt interactions, are used in cosmetics, in straightening hair or curling hair. How is hair curled? By adding chemicals and heat. How is hair straightened? By adding heat and chemicals. What is hair? Hair is a protein. These chemicals that we use in our hair to either straighten or curl the hair will disrupt some disulfide bonds and make others form. They will disrupt some hydrogen bonds and make others form. So they will change the shape of the keratin protein of our hair, and our hair can go from straight to wavy, or from wavy to curly, or from curly to wavy, or from curly to straight. So protein structure, the way proteins are held together, the structure of proteins, is used every day in everyday life to manipulate hair. Now, I want you to look at this picture and ask yourself, what is happening here? I want you to pause the video, really pause it and think. And once you have the answer, you can unpause the video and see whether we're on the same page. So what you're seeing here is a folded protein. It is in 3D shape that is now partially, or you can even say completely unfolded. So this is the denatured protein. The protein is denatured. It has lost its shape. Same thing happening down below, but now this arrow here shows that this denatured protein can actually fold back into its native shape. So for many proteins, as a matter of fact, for most proteins, if you heat them, let's say you boil them, and you let them cool down very, very slowly. So you're in a lab, in a test tube, you have a protein, you have an enzyme. You boil the enzyme, you boil the protein, and then you let it cool slowly. As the protein cools, it will refold back into its native shape. Think about what this is evidence for, this particular experimental result. So you can denature a protein, and then you can renature it. You can let the protein be, and the protein will refold to its native state. And we're going to talk about this more, but I want you to think about, what does this mean? What can this be used as evidence for? What can this be used evidence for? The hint I'm going to give you is, what determines the structure of this protein? What determines how the protein is going to fold? And this is the end for proteins, my friends. So I'm going to see you next time when we talk a little bit about nucleic acids. Goodbye for now.